This is part 2 of the recorded lecture on viruses, and this is where we left off with this slide showing a host cell, which is a human cell. And it's showing a virion, which is, remember, that's just the name for an individual virus particle. And this particular virion has its nucleic acid core, and then it's got a protein capsid, which looks to be some kind of polyhedral shape, and then it has an envelope, and then attached to the envelope are these little green glycoproteins. So this particular virus, the KSHV virion, uses glycoproteins, and those glycoproteins attach to the viral receptor in the host cell. And the host cell viral receptor in this case is called the XCT receptor. And you can see here how it's got a special shape, but especially right in here, it's shaped to where the viral receptor can fit into that shape. And then we have two other molecules that have to bind to a CD98 and an alpha 3 beta 1 molecule. So these are, I would say, they assist the binding process, the attachment process, and they sort of provide an extra, they provide a way for the virus to make sure that it's attaching to the correct host cell, if that makes sense. So this shows the attachment stage of a viral cycle, a viral cycle of attacking a host. Now, what we're looking at here is a T4 bacteriophage, which is a virus that infects an E. coli bacterial cell. And this is just showing you an illustration of how viruses can have a complex shape. So the head, in fact, the head is the capsid in this virus. And that capsid is a naked icosahedral shape. And the tail is actually helical shape, but it's got a lot of branches. It's got what looks like little legs that are attached to the tail. And they even appear to be designed for attachment to the host cell. So having an icosahedral head and a helical tail means that we call the shape complex. And by the way, the T4 bacteriophage is a DNA virus. Now we've got another shape, which I'm going to show you. This makes it look like it's kind of an irregular shaped ball, but it's actually got a geometrical shape to it. So that illustration on the left is actually what the adenovirus looks like. So an adenovirus is a naked virus, means it does not have an envelope. So what we're looking at is the DNA core. The individual capsomeres are these little kind of brown ovals, and the capsomeres together make up the capsid of the virus. And then the glycoproteins are spikes, which are used for attachment to the host cell. And here you see an illustration of an adenovirus, and it gives you some more specifics. We knew it was a DNA virus, but it is actually a double-stranded DNA virus. It's transmitted orally, and it causes a variety of illnesses in vertebrates, including human eye and respiratory infections. So an adenovirus is a naked icosahedral shape. Here is an image of an enveloped icosahedral virus. and that's HIV. This is HIV. HIV is also a retrovirus. So what have we got here? We've got an RNA core, and here you can see that the RNA is divided into segments. So it's actually segmented. And with it being a retrovirus, the RNA core is actually converted to DNA. And that's the opposite of transcription. So it's the reverse of transcription. RNA is converted to DNA by reverse transcriptase, and then the DNA is converted into messenger RNA for transcription, and the messenger RNA undergoes translation to produce the protein. You can see that there is a capsid which has an icosahedral shape. You may not be able to tell that from looking at it, but it does. And then there's an envelope outside the capsid, and there are glycoprotein spikes which help in attachment to the host. Here we have two viruses, the rabies virus, and then variola virus is the virus that causes the disease smallpox. So a rabies virus is an example of a single-stranded RNA virus with a capsid and an envelope. And then the variola virus is a double-stranded DNA virus with a capsid and an envelope. And if you've never seen what smallpox looks like, because we very rarely have, and smallpox has been eradicated in our world. And so, thank goodness we don't see this. We don't get to see people with smallpox like they did years ago. All right, so viruses can be classified according to their nucleic acid core and also according to their structure. So some things that we look at are whether the nucleic acid is RNA or DNA, and then whether it's single-stranded or double-stranded. So I'll give you an example. If we look here at this list, the rabies virus and the retroviruses, we can see that they are single-stranded RNA viruses. So you can actually describe it as single-stranded RNA. And then the herpes virus and the smallpox virus could be described as double-stranded DNA. And then when you put all four together, all four are linear. So I could actually add that rabies and retroviruses are linear single-stranded RNA viruses, and the herpes viruses and smallpox viruses are linear double-stranded DNA viruses. And then you have some examples of viruses that have circular nucleic acids. Bacteriophage, this is not a surprise because bacteriophages are viruses that infect bacteria, and the chromosome in a bacterium is a single circular chromosome. So it's the same way for the virus that infects a bacterium. Another virus that's listed here is a papillomavirus. You've probably heard of HPV, which is human papillomavirus. Human papillomavirus causes genital warts, and there is a vaccine for that children can get starting at age 11. And it will prevent them from contracting HPV and genital warts. And then what that does is prevents women from getting cervical cancer later in life, because human papillomavirus can cause cervical cancer later. Some viruses that have a non-segmented genome, in other words, the genome is just a single segment of genetic material, are parainfluenza viruses. And then segmented genome, the influenza viruses which cause the flu are segmented. They have more than one piece of nucleic acid. Now, the capsid structure is described as naked or enveloped, and then you say whether it's icosahedral, helical, or complex. So some examples of naked icosahedral the adenoviruses, and that was the shape of virus that we saw earlier that causes that is transmitted orally and can cause some um, eye infections and respiratory infections. But another example of a naked icosahedral virus is hepatitis A and also poliovirus. Then enveloped icosahedral, we looked at the HIV. Epstein-Barr virus is, gosh, I forget what it called, mononucleosis is Epstein-Barr virus is the one that causes, I remembered it was something common, but it's the one that causes mononucleosis. We used to call that the kissing disease, but it's very easy, it's very uh, easy to catch. So enveloped helical viruses, helical shape would be like the tobacco mosaic virus, but basically when we looked at the tobacco mosaic virus back in the beginning, so these look like sticks, but really, and my hands are shaky, but really inside of that is a helical structure. So it's kind of like DNA. It's spiral shaped. Okay, so that's what is meant by helical. And so the tobacco mosaic virus is naked helical, but the influenza that causes the flu, the mumps, the measles, rabies virus are enveloped helical. And then we have some examples of complex, which means they're combinations of icosahedral and helical structures. The main example we have looked at thus far is T4 bacteriophage. And here you see images of the different shapes. A is naked icosahedral, like the adenoviruses. They look circular, but they're actually more of a geometrical shape. And then B is enveloped icosahedral. So here's the capsid in here, and then here's the envelope outside. Your HIV is an example of that. This is enveloped helical. So it's kind of like an envelope around one of these. And then D is naked helical, and E is a complex shape. It's kind of hard to look at E and tell what it's supposed to look like. So that's okay. Now what we're going to do is we're going to review protein synthesis in cells, because when we talk about the Boston classification system, we will be looking at how messenger RNA is produced. In other words, how transcription occurs. So in living cells, we learned in Bio 111 that double-stranded DNA is used to produce a strand of messenger RNA, and we call that process transcription. The messenger RNA is single-stranded and the DNA is double-stranded. And then that messenger RNA, which is produced using the enzyme RNA polymerase, is used to code for the individual amino acids in the protein. Remember, amino acids are the building blocks of proteins. So messenger RNA has the code for each amino acid, and that is called translation, when the messenger RNA codes for the actual protein. So we know that viruses have to make proteins, and so the Baltimore classification focuses on the mode of messenger RNA production, or in other words, it focuses on the process of transcription. There are 7 different groups of viruses, and I'm going to stop here because we're at our 15 minutes, and this is where I will begin